Hello, this is Team 7's project on redesigning melanoma detection by Julia Braz, Kieran Wallace, and myself, Sydney Steele. It is estimated that 1 in 5 Americans will develop skin cancer in their lifetime, and more than 1 million Americans are living with melanoma. Currently, the best diagnosis protocol to detect skin cancer is via biopsy. This is an invasive process that includes the removal of skin from a patient's body for laboratory examination. Results can take anywhere from several days to a few weeks, and multiple appointments are needed afterwards to discuss results, diagnoses, and treatment options. There are also many risks that come with biopsy procedures, including bleeding, bruising, scarring, infection, or allergic reaction to topical antibiotics. Our goal of this project is to create a sensor system to find a less invasive method of detecting skin cancer, specifically melanoma, through engineering HEC293 T cells and utilizing a designed synthetic notch pathway to sense CD44 in the HT144 cell line. To kick off our experiments, we begin by designing a very specific sensor cell. The key component to our engineered plasmid is a synthetic notch pathway. The pathway is user-defined to drive a range of functional responses in different mammalian cells. There are 3 main parts of our SynNotch pathway: the recognition domain, SynNotch protein, and transcriptional factor/intracellular domain. When this process occurs, the Notch protein turns on and cleaves from the anti-CD44 single-chain variable fragment. The Notch protein is a direct transmembrane receptor that contains a transcriptional regulator released from the membrane when an extracellular ligand induces intramembrane signaling. This cleaves Gal4, our transcription factor domain, a responsive promoter driving the expression of mCherry, a red fluorescent protein inside of the sensor cell.
Incorporating our designed SynNotch pathway, we constructed our entire DNA plasmid, beginning with a start codon and signaling sequence. The signaling sequence enables proteins to find the correct location outside of the cell membrane by tagging them. Following is the MYC tag, a polypeptide protein tag that helps to detect the expression of recombinant proteins. Next is our SynNotch pathway. We then inserted the sequence of DNA into the pTwist CMV WPRE Neo HEC293T expression vector. The CMV-driven expression vector is designed to deliver high levels of transgene expression. This vector also contains WPRE that enhances transgene expression. After designing and ordering the plasmid DNA sequence, we obtained a glycerol stock of Rogelio's RHLB050 DNA. At this point, we obtained both our designed twist vector plasmid DNA and the parental vector DNA. The first step is retransformation, a process of incorporating genetic material into a cell through its membrane. We retransformed both DNA plasmids into E. coli separately, and then streaked both E. coli colonies onto separate LB anti-streak plates and incubated overnight. The following day, we chose one individual colony to mini-prep. DNA mini-prep is a procedure used to purify plasmid DNA from bacterial cell cultures. Then we began the transfection process of both Rogelio's RHLB050 DNA and our designed twist vector plasmid DNA into HEC293 T cells. Since the transfection process was the biggest risk of our experiment, a fluorescent tag, BFP, was embedded within the glycerol stock DNA in order to measure whether the transfection process was successful or not.
Since our plasmid DNA is not tagged with fluorescence, we performed flow cytometry, which is a technology used to analyze single cells as they pass through lasers. Implementing our McTag protein within our plasmid, we introduced a c-Myc monoclonal antibody to our transfected cells so that the flow cytometer could recognize whether the transfection was successful or not. Unfortunately, our results from the flow cytometry did not detect that our McTag protein or BFP was present in the transfected cells. This means that something went wrong with our first trial of transfection. Based on our flow cytometry results, we decided to transfect a new 6-well plate of HEC293 T cells in an effort to have a more successful transfection. We did not have time or resources to perform flow cytometry again on these newly transfected cells, so we went ahead with the experiment in hopes that it was successful. We then made a co-culture by plating a 1-to-1 ratio of our target cells to sensor cells. Our target cells are the HT144 cells, which are a malignant human melanoma cell line that expresses the surface antigen CD44. Our sensor cells are the HEC293 T cells that we bioengineered to detect the CD44 and express mCherry, a red fluorescent protein, when present. 24 hours after our co-culture, we did our first round of imaging. We initially did not see any presence of mCherry on either the first or second trials of co-culture. However, after 48 hours, we were able to detect some mCherry in our. Although our first transfection did not work, these results show that our second transfection was successful because mCherry was expressed on our imaging of the second trial of transfection and co-culture, showing where the sensor cells detected CD44 in the target HT144 cells. The results from our project show that it is achievable to design a working sensor cell that detects CD44 in human melanoma. This is an exciting start to a larger goal that aims to eliminate invasive skin cancer detection with bioengineered sensor cells. Thank you for listening.